the shared neural fingerprint. How psychedelics reshape brain connectivity, a groundbreaking large-scale study has identified a consistent neural fingerprint left by various psychedelic substances on the human brain. By analyzing hundreds of brain scans, researchers have discovered that while different psychedelics, such as LSD, psilocybin, DMT, mescaline, and ayahuasca have unique qualities, they all trigger a remarkably similar pattern of brain activity. Breaking down the hierarchy, the core finding of the study, published in Nature Medicine, is that these substances fundamentally alter how the brain's internal systems communicate. Under normal circumstances, the brain operates through a structured hierarchy, where certain higher-order networks manage complex thought and decision-making, while lower-order networks handle basic sensory input like vision and touch. Psychedelics appear to flatten this hierarchy. Instead of a controlled, top-down flow of information, the drugs induce what researchers call excessive crosstalk. This results in unleashed communication. Brain networks that usually operate independently begin to communicate wildly with one another. Sensory integration. Higher-level cognitive networks become intensely linked with primitive networks responsible for sensation and vision. Dissolution of self. This breakdown of traditional boundaries may explain the subjective experience of ego dissolution, where a person's sense of individual identity seems to merge with the world around them. A new scale of scientific rigor for years, the scientific community has struggled to draw definitive conclusions about psychedelics because most studies were limited by small sample sizes. This lack of data made it difficult to distinguish between the effects of a specific drug and the general experience of a trip. To solve this, researchers conducted what is believed to be the largest study of its kind to date. They synthesized 11 different imaging datasets from 5 countries, analyzing over 500 brain scans from 267 participants. This massive dataset allowed scientists to move past anecdotal evidence and observe a shared biological mechanism. Notably, the study also clarified a common misconception. While the communication between networks changes, the networks themselves do not disintegrate or fall apart. Rather, they simply lose their usual boundaries. Why this matters for modern medicine, the timing of this research is critical. As the psychedelic renaissance continues, these substances are being aggressively tested in clinical trials as potential treatments for treatment-resistant depression, post-traumatic stress disorder, PTSD, schizophrenia, and other neurological conditions. For these drugs to move from experimental settings to mainstream medical practice, they must be understood with precision. As Dr. Danilo Bzdoc of McGill University noted, much of the existing research has been built on shaky ground. By identifying this shared neural fingerprint, scientists are providing the solid foundation necessary to develop safe, standardized, and effective psychedelic-assisted therapies. If psychedelic research is to mature responsibly, it needs large-scale, coordinated evidence. Dr. Emmanuel Stamatakis, University of Cambridge conclusion by uncovering a universal pattern of excessive crosstalk. Across different psychedelic substances, researchers have moved the field from anecdotal observation toward a rigorous biological understanding. This discovery provides the essential framework needed to safely integrate psychedelic therapies into modern mental health care.